Friends, today we are going to discuss the sex determination process as explained by Correns. It is also known as the sex chromosomal sex determination. And after this, we are also going to discuss the sex determination process in human beings. Let us get started. Sex determination is a process by which the embryo of an organism develops into an adult of a particular sex. What is the actual process by which an embryo becomes a male individual or an embryo becomes a female individual is called the sex determination process. It is actually a biological system that forms the secondary sexual characters, primary sexual characters separately in males and in females. Several factors influence the process of sex determination. Not only the sex chromosomes, not only the genes. So in some animals, sex chromosomes are playing a very important role. In some animals like Drosophila, genes are playing a very, very important role. And certain environmental factors are also there to determine the sex in certain animals. Hormonal factors are there and metabolic factors are also determining the sex. Let us see the different phenomena. Let us see the earlier concepts of sex determination. Aristotle, 335 BC, opined that heat of the body of father during copulation will determine the sex of the embryo. If father is having more heat, then more chances of male offspring be born. So he also advised certain people to conceive during summer. So if any individual conceives or any mother conceives during summer season, more chances of male hairs for them, but it is totally wrong. It is unscientific. There is no question of heat of the body of father will determine the sex. This is totally blunder and wrong. Galen in the 200 BC, women are nothing but men with some changes in the arrangement of their genitalia. He was the first person to explain that the reproductive organs of both males and females are homologous organs. Okay, so we see that man and women are not having much differences in all other characters, and even in males also, uterus masculinus, a small uterus is there. So womb man might be converted into woman in gradual cultural development. Andreas Vesalius in the year 1543 observed female genitalia like structures in the male genitalia. Female genitalia like structure, that means there is a very small uterus. Okay, uterus masculinus like that. So these scientists try to explain that there are no much differences in the structure of the reproductive organs of males and females. But for the first time, a scientist by name Regnier de Graaf, first time explained and identified or reported that women are going to produce small eggs. And these eggs, which are liberated into the body cavity coelom, are responsible for transmitting the traits from parents to the offspring. As he was the pioneer of these observations, Regnier de Graaf is considered as the father of reproductive physiology. Okay friends, these are some earlier concepts, but what are the actual factors of sex determination? What actually determines the sex of the embryo? Okay, so whether it is the food that the parents are taking? No. The food what parents take will not determine the sex, because some superstitions are there. Some superstitions are prevailing in our society, so we need to understand. So if when mother is conceiving a baby, if mother takes a variety of food, then a particular sex may be born. That is totally wrong. Time of the delivery does not influence. Time of copulation does not influence. Is there any astrological belief? That is totally wrong. Habits of parents don't determine the sex. But what are the scientific concepts of sex determination? Let us see. So, from 1896 to 1901, some very important reports came with reference to the sex determination. Henking, McClung, they were working on these diptera or locus insects, certain insects. So, they identified during the meiosis of these insects, the sperms are having unpaired chromosome. In some sperms, there is unpaired chromosome. Perhaps this unpaired chromosome is determining male sex. It is for the first time Henking and McClung noticed. So Henking named it as X body or X element. So later it is named as X chromosome. So for the first time, these two scientists explained about the X chromosome in the sperms of locust. Mendel laws were rediscovered by Hugo de Vries, Karl Correns and Erich Schemark. Because of these uncovering or rediscovery of Mendelism, many scientists were interested in working in genetics. So by that way, a lot of research is done on the topic of sex determination. Stevens and Wilson identified there is a very, very small chromosome associated with that unpaired X chromosome in males. That small chromosome they termed as Y chromosome. By this way, X chromosome and Y chromosome are identified, and it is very well known by that time that the X chromosome and Y chromosome are very, very special chromosomes, and they are bringing male characters like that. Sex determinating characters are there with the X and Y chromosomes. So, scientists have come to a conclusion that chromosomes can be separated into autosomes and allosomes. Okay. Correns explained the process of sex determination in the most simple and very convincing manner. He explained sex chromosomes like X and Y are responsible for the sex determination in the embryonic development. So, he was the first person to explain the sex determination process in a scientific way, which is very, very simple to understand even to a layman. Okay. Balzer explained that in Borrelia viridis, environmental factors are playing very, very important role. Environmental sex determination. Okay. Next, Calvin Bridges explained in Drosophila, there is no role to Y chromosome to play. There is no role of Y chromosome to play in male determination in Drosophila. So he proposed a balance between X chromosome and autosomes. So this balance theory is called genic balance theory. We are going to discuss the genic balance theory in the next video. Okay. F.R. Lilly, another scientist, explained the influence of hormones. So he was the first person to explain about free martins in cattle. Riddle explained the metabolism role in the sex determination in certain insects. In the year 1938, a doctor by name Turner observed 7 women who are not having women characters perfectly. It's because they didn't have 2 X chromosomes. As in general, women are having 2 X chromosomes, males are having only 1 X and 1 Y chromosome. But Turner for the first time observed that some women are possessing only 1 X chromosome. So such women who are not developing full women characters or feminine characters are reported as Turner's syndrome, and this is because of nondisjunction of sex chromosomes. In the year 1942, Klinefelter, another doctor, reported that 9 men are having an extra X chromosome. So, they are males but having extra X chromosome. And such individuals who are having 2 X chromosomes and 1 Y chromosomes are named as Klinefelter syndrome. So, these kinds of reports created a lot of interest among scientists to work on the actual procedure of what is that called sex determination. Murray Barr, Bertram, and other scientists identified Barr bodies in female cells. Mary Lyon explained that Barr body presence is because of extra X chromosome. That means out of 2 X chromosomes in females, one is subjected to dosage compensation or heterochromatinization or inactivation. So they are the scientific explanations on Barr bodies. Jacobi scientists reported sex reversal in fowls. The female fowl after laying the eggs and completing its cycle of laying eggs turned to male. This is called sex reversal. And Charnier and other scientists explained environmental factors in sex determination in several patterns. Pattern A, where low temperature brings the maleness. Pattern B, where high temperature brings the males, or low temperature makes females. And pattern 2, extreme temperatures. When eggs are incubated in extreme temperatures, they become males, and when eggs are incubated in medium temperatures, they became males. Extreme females and medium temperatures males. These are all some kinds of reports related to sex determination, and let us concentrate on certain terminology to understand the process of sex determination. Okay friends, autosomes. Autosomes are nothing but 22 pairs of chromosomes in human being. And these 22 pairs of chromosomes are having loci for the genes which regulate our body characters. Our bodily characters are expressed by the genes located on autosomes. Autosomes are responsible in human beings. Autosomes are responsible for producing or expressing or performing bodily characters. Of course, these autosomes in the case of Drosophila are also determining male sex. Okay, allosomes. Allosomes are the sex chromosomes. They are XX in females and XY in males. They are having the genes for determining the sex. And they are having certain genes which are going to develop gonads. The gonads either to be converted into female ovaries or to male testes. So allosomes are having those particular genes. So friends, autosomes, allosomes, we need to understand, we need to have this meaning of these terms. And homogametic parent, the parent who produces similar type of gametes with reference to allosomes are called homogametic. For example, in human beings, the female mother produces eggs, and all the eggs are having one X chromosome equally. So all the gametes of mother are same. There is no difference in allosomes. So that's why mother is called homogametic. What I am speaking are related to X and Y chromosomes only. Heterogametic parent is that parent who produces two different types of gametes. You take for example in human beings, father produces sperms, but sperms are of two kinds. Some sperms are having X chromosome and some sperms are having Y chromosome. Or in other animals, some are having X chromosome, some are not having X chromosome. In that way, sperms are of two types. So whenever father or any mother or father produces different kinds of gametes, such a parent is called heterogametic. If mother or father produces similar type of gametes, such parents are called homogametic parents. Okay, next term, primary sexual character. Primary sexual characters are those characters which appear from birth onwards. Say, presence of gonads. For example, ovaries in females, testes in males. And the external genitalia of females, the external genitalia of males, they appear from birth onwards. Second, they are determined by chromosomes or genes. Secondary sexual characters are those morphological changes that appear whenever the body attains sexual maturity. For example, if boys attain sexual maturity, they develop some special characters which are not there in females. If girls attain the sexual maturity, there some morphological characters, some morphological changes appear. So such characters are called secondary sexual characters. And secondary sexual characters are expressed by hormones, sex hormones. Okay. Sex determination. Sex determination is a procedure by which the embryo becomes male or female. It is by genetic nature. Sex differentiation. Sex differentiation is only a phenomenon with which we can differentiate the sex. You are all males, you are all females by observing the external characters, secondary sexual characters. Okay. Sometimes sex differentiation may be wrong. You take, for example, the voice of boys will become low pitched. If it is not converted, we feel that a girl may be speaking like a boy and we may feel that the girl as a boy. So this is not correct to determine the sex. This is only a differentiating external separation of males and females. Right. So these are certain terms we need to have in our vocabulary to understand sex determination process. Haploid status. Having only one set of chromosomes is called haploid. Okay. And you take, for example, in human beings, we have 46 chromosomes, that is diploid. If our cell is having only 23 chromosomes, that is called haploid. Diploid means having two sets of chromosomes. So, in addition to haploid and diploid, there is a very special condition called aneuploidy status. Aneuploidy status is a very, very special status, maybe occur because of mutations. And in this status, we have the diploid number of chromosomes, but one chromosome is extra, or two chromosomes are extra, or one chromosome is less, or two chromosomes are less to the total number. If the total number is 46, sometimes a person may be having only 45 chromosomes. That means only one chromosome less to the total number of chromosomes. So, not the entire set changes here, only one chromosome number changes. So, such a condition is called aneuploidy, and it may be 2n. 2n means diploid number, minus 1 chromosome, 2n minus 1 monosomy, 2n plus 1 trisomy, 2n minus 2 nullisomy, 2n plus 2 tetrasomy. 2n plus 1 means we have 47 chromosomes instead of 46. 2n minus 1 means the person will have 45 chromosomes instead of 46. So, like that, tetrasomy 48 chromosomes, nullisomy 44 chromosomes. Okay, one more word is very, very important. Disjunction. The two homologous chromosomes tended to separate. The two homologous chromosomes, what are there in our body? The first pair is having two homologous, second pair is having two homologous, third pair is having two homologous. So, two homologous chromosomes tend to separate during meiosis. This is called disjunction. If anything happens and the two homologous chromosomes never separate, failure of separation of two homologous chromosomes is called non-disjunction. Because of non-disjunction, Klinefelter syndrome, Turner's syndromes are formed in human beings, and lot of sexual aneuploidies are generated in the case of Drosophila as observed by Calvin Bridges. Okay. So, in how many ways are there for sex determination? What are the different methods of sex determination in animals and plants? So, sex determination can be done by sex chromosomes XY. Sex determination in Drosophila occurs with the help of a balance, that balance male genes and female genes, and whose load is heavier, and that will become that particular sex. This is called genic balance theory. Haplodiploidy, it's a condition we will see in certain Hymenoptera insects like honeybees, ants, sawflies, etc. And in some bacteria and in certain plants, there may be a single gene effect for determination of a sex. Okay friends, today's lesson, let's concentrate on Correns theory of sex determination. Correns theory of sex determination. Let us concentrate on this particular theme today. Correns explained that sex chromosomes are responsible for the determination of sex of embryo. Females and males are having different sets of sex chromosomes. Because of these different sets of chromosomes, the embryo becomes a particular sex. Embryo becomes an adult of a particular sex. Mainly, Correns theory explains that the parent who is heterogametic, the heterogametic parent is determining the sex of the embryo. So this is also known as heterogametic sex determination. Parents are two types. A parent is a homogametic because he or she produces similar type of gametes with reference to allosomes, and another parent is heterogametic. The gametes are different with reference to the sex chromosomes. So, homogametic parent never determine the sex of the embryo, but a heterogametic parent will determine the sex of the embryo. Okay, so this method is broadly, Correns theory of sex determination can be broadly divided into two headings: heterogametic males, heterogametic females. Heterogametic males, heterogametic females. In certain cases, when the sex chromosomes are not same, then they will become males. In certain other cases, when sex chromosomes are not same, they will become females. Heterogametic males, heterogametic females. Let us understand this by observing this chart. Okay, these two are heterogametic males and these two are heterogametic females. Let us understand this theory very clearly. Let us first see heterogametic males. Friends, you might have understood what is the meaning of heterogametic males. Heterogametic males means the male is going to produce two different kinds of sperms. In this case, right side. Heterogametic female means the female is going to produce two different types of eggs with reference to sex chromosomes. Sex chromosomal content is different in different gametes. Okay, first let us study heterogametic males. Correns explained heterogametic male sex determination in two methods. One is called XXXO method. The other one is called XXXY method. XXXO method, XXXY method. Okay, XXXO method is observed in grasshoppers and in locusts. In locust and grasshopper like insects, we observe XXXO type. What is XXXO type? XX is the female, XO is the male. So, how is the female? Female is having two similar type of sex chromosomes, XX. So, female is called homogametic. And you see the male is having XO, that means one X is there, there is no other chromosome. So, male is heterogametic. Female is homogametic. Let us see, a female produces similar type of gametes. A refers to autosomes because autosomes are common both in males and females. I need not mention, autosomes are not different. They are same in males and females. Let us see how is the female going to perform the gametes. Here the female is going to produce AX gamete and AX gamete. Both are same. So homogametic. And you see the male individual, male parent. Male parent is going to form sperms of two different kinds. Some sperms are provided with X chromosome and the rest of the sperms are not provided with any X chromosome or Y chromosome like that. In 50% of the sperms, okay, number of the sperms, there is X chromosome, and in the rest of the chromosomes, there is no chromosome at all. 0 means no chromosome. Okay, now let us see, when an egg is fertilized, let us consider this egg got fertilized. When egg is fertilized, it will develop the female and it can also develop the male. So there is no role of sex determination. An egg can form, an egg can develop either into a female or into a male. You see this egg. This egg can develop either into female or into a male. So there is no role of sex determination with the eggs. Now let us consider the sperm. If the sperm with X chromosome is fertilized, definitely females are born. The embryo becomes a female only. If the sperm without X chromosome is fertilized, definitely it will become male. So which is having the sex determination? The sex determination capacity is there with the sperm that is going to fertilize. And one very important thing is that at the point of fertilization, the sex is determined. So the point of sex determination is fertilization. Before fertilization, we cannot say what embryo is going to be born, what embryo is going to be developed, whether male or female. At the point of fertilization, we can determine the sex of the embryo. So it is, in this case, it is the sperm that is going to form the sex, determine the sex. This is called XX, XO method, example grasshopper and locust. And let us see another heterogametic male method. This is called XX, XY method. Here, mother female is XX, father male is XY. Let us see mother produces the eggs of same kind, X, same kind. But father produces the sperms of two different kinds. Some sperms are provided with the X chromosome and some are provided with the Y chromosome. If egg is fertilized, it can form either female or male. If a sperm with the X chromosome is fertilized, definitely females, embryo becomes a female. If a sperm with the Y chromosome gets fertilized, definitely the embryo becomes male. So, the sex determining capacity is there with the sperm that is going to fertilize. And XXXY method is exemplified in human beings and in Drosophila by Correns. Okay, of course in Drosophila we are going to discuss in the next lesson the genic balance theory, but it looks like as if it is following XXXY method. Okay friends, these are heterogametic sex determination, male sex determination. Let us see heterogametic female sex determination. Heterogametic female sex determination according to Correns is of two types, ZOZZ, ZWZZ. Friends, you need to understand here Correns used Z, a letter Z instead of X. Z is not a separate chromosome. Here it is representing an X chromosome. W is not a different chromosome, that is representing Y chromosome. To avoid confusion, only the letters are changed here. Okay. Now, ZOZZ type. ZOZZ type is observed in a moth called Fumia. Where female is ZO, that means heterogametic. Male is ZZ, same chromosomes are there, that is homogametic. Quite contrary to the earlier method where males are heterogametic, but here females are heterogametic. Okay, let us observe the sperms formed by the male. The sperms produced by the male are of same type, AZ. But the eggs formed by the female are two types. Half of the number of eggs are provided with the Z chromosome probability and the rest are without Z chromosome. Okay, let us see a sperm. If a sperm is fertilized, it can form either male or female. If a sperm is fertilized, it can form an embryo of male or a female. So there is no capacity of sex determination with the sperm because both the sperms are same. If an egg with the X chromosome, that means Z is written here, if an egg with the X chromosome is fertilized, definitely male individuals, embryos become male. If the egg without X chromosome, that means Z chromosome, is fertilized, it will become female. That means embryos determine female sex. So friends, determination capacity is there with the heterogametic females. This is called ZOZZ. Example, Fumia. Okay, let's see the other method explained by the last method explained by Correns. This is called heterogametic female method, ZWZZ method. Here Z represents the X chromosome, W represents the Y chromosome. Females are heterogametic with Z and W chromosomes, X and Y chromosomes. Males are with ZZ, XX chromosomes. Male produces the sperms of same kind, AZ. but female produces the eggs of two different kinds. One egg is with AZ, one means 50% almost all, and the rest of the eggs are with W chromosome. I'll mention once again, Z means X, W means Y. Okay, if a sperm is fertilized, it is going to form any sex. It can be formed any sex. But if an egg with Z chromosome is fertilized, definitely male individuals, embryo grows into male individuals, male embryos. If egg with W chromosome, that means Y chromosome is fertilized, then definitely the embryos will be the female embryos. And such kind of determination by heterogametic females, ZWZZ, occurs in certain birds, reptiles, and fishes. Okay friends, let us summarize the Correns method of sex determination. Correns method of sex determination depends on heterogametic individual parents. If a heterogametic individual is a male, that is called heterogametic male method, male heterogamety. And if it is the female, female heterogamety. Male heterogamety two types, XXXO, XXXY. Female heterogamety is of two types, ZOZZ and ZWZZ. Okay, these are the examples. So this is a very simple and most convincing theory of sex determination by Correns. Okay friends, let us observe how the sex of the human being is determined. Okay, now my question is, what do our parents give us? Our parents, what from our parents we get? Of course, there is no difference in autosomes. We get 22 pairs of autosomes, 22 autosomes from father and 22 autosomes from mother. There is no difference. But coming to the allosomes of mother, allosomes of father, how do they transmitted? How do they go to the next generation? How are they transmitted? Okay, so let us consider here mother. Friends, you know very well, mother has two X chromosomes. I have written these two X chromosomes, X1, X1, only to say where is it going. I numbered it as X1, X1. Now let us see, and father chromosome is X2 chromosome. Actually, we have to say X1, X2 in mother and likewise. But to say where the X chromosome of mother is transmitting to which progeny, so I have given the numbers only for the observation. Let us see. Mother's X chromosome goes to son and also goes to daughter. Okay, mother can transmit her X chromosome either to son or to daughter. Okay, in other words, I say daughter receives one X chromosome from mother, and the X chromosome what is there with the son. The son's X chromosome is definitely mother's X chromosome. Okay, son will get the X chromosome, or son is inherited X chromosome only from his mother. So son gets X chromosome from mother. That is the very important thing we need to understand. So by that X chromosome, lot of diseases from mother to son occurs. We observe that in crisscross inheritance, as in the case of color blindness, or in the case of hemophilia, or in the case of Duchenne muscular dystrophy, and we prepare a next lesson for that to understand that process. So the very important concept is that son receives X chromosome from mother only, but daughter receives one X chromosome from mother and one X chromosome from father. Daughter has two X chromosomes, one X chromosome from mother and one X chromosome from father. Now the question is, to whom father will inherit his X chromosome? Father's X chromosome goes to daughter only. Father will never give his X chromosome to son. Give here refers to inheritance. So X chromosome of father will never be inherited to son. X chromosome of father will definitely be inherited to daughter. Okay. Then what about the Y chromosome? Y chromosome which is there in father, it's a male chromosome actually. Y chromosome is a male chromosome. So it is present only in the male pedigree. So Y chromosome from father definitely goes to son. Okay, Hollandric pathway we say. So this slide will give you certain concept of how the X chromosomes of son and daughter are inherited. Okay, sex determination in human beings, let us see. And for the sex determination, as I have explained in the Correns theory, it is nothing but XXXY method. Okay, XX is mother and XY is father, and mother produces the two eggs with X and X. Father produces two kinds of sperms, one with the X chromosome and one with the Y chromosome, and the sperm that is going to fertilize will determine the sex. If the sperm with the X is fertilized, male embryo turns to male. If sperm with the X chromosome is fertilized, embryo turns to female. If the sperm with Y chromosome is fertilized, embryo turns to male. Let me repeat. If the sperm with X is fertilized, female. If the sperm with Y is fertilized, male. Okay. So sex determination can be done in the cells also. If two cells are given, how are you going to determine the sex of the two cells without properly observing the chromosomes? It is very easy. So Murray L. Barr and Bertram, Keith Moore and others found that the female cell has a black structure. A strained structure is there, and that strained body is called Barr body. Whenever Barr body is present, we need to understand that there is more than one X chromosome. That means one Barr body, actually Barr body is nothing but out of the two X chromosomes of female, one X chromosome is subjected to inactivation or heterochromatinization. Because of heterochromatinization of one X chromosome out of two X chromosomes in female, the heterochromatinized X chromatin appears as a stained body. That body is called Barr body. So what is the indication? When Barr body is there, we can say that it is female. When Barr body is there, it is female cell. When Barr body is absent, we can say that it is male cell. But how to count the number of Barr bodies? Number of Barr bodies is equal to number of X chromosomes minus 1. If two X chromosomes are there, 2 minus 1, 1 Barr body. That is female. If one X chromosome is there in males, 1 minus 1, 0 Barr body in males. And why X chromosome is subjected to inactivation? X chromosome is subjected to inactivation only to compensate the dosage of sex chromatin in male and female. In females, double the sex chromatin is there when compared to the amount of sex chromatin in males. That's why in females, sex chromatin is subjected to heterochromatinization or inactivation. Okay, this is called dosage compensation or heterochromatinization. We can observe the heterochromatinization in the blood of females also. In the blood of females, the WBC, especially the neutrophils. In neutrophils, if we see under a microscope, the nucleus of neutrophils is possessing a small structure, drumstick-like structure, that is called drumstick body, which is nothing but a Barr body. So the blood which exhibits a drumstick body in the neutrophils is female blood. The blood where the neutrophils are not having drumstick body is said to be the male blood. Okay. Now, sex determination in humans, we'll see with the number of chromosomes. Father has 44 autosomes, 2 X chromosomes. Mother has, Mother has 44 autosomes, 2 X chromosomes. And father has 44 autosomes, X and Y chromosomes. And what are the eggs produced by mother? So eggs produced by mother are having 22 autosomes plus 1 X, 22 autosomes plus 1 X. That means eggs are same. But let us see the sperms. The sperms here produced by father, half of the number of sperms are provided with X chromosome, and the rest of the sperms are provided with Y chromosome. Okay, if the sperm with X chromosome is fertilized, it will become female. If the sperm with Y chromosome is fertilized, it will become male. How the male is formed? Because the Y chromosome is having a region. Small Y chromosome is the sex determining chromosome in males. No matter how many X chromosomes are there, if one Y chromosome is there, the embryo becomes male. Okay, this is the difference between a man and a Drosophila with reference to the functioning of Y chromosome. In man, Y chromosome definitely determines male sex. In Drosophila, Y chromosome has no role to play in sex determination, but it is responsible for sperm production. Okay, so why brings maleness? This is because the small Y chromosome is having a very small region called SRY region, sex determination region on Y, SRY, sex determination region on Y. That region produces a factor called a testis determining factor, and because of that, whenever Y is there, testes are formed. Whenever testes are there, males are born, or the embryo turns to male. So in this method of sex determination, there is equal chance of formation of male and female. So in this method of sex determination, there is no discrimination that more males can be born, more females. No. Male determination and female determination is same. So the sex ratio by this method always 1:1. Sex ratio is always 1:1. Okay. And sometimes what happens, this is a very, very interesting thing to note. Sometimes the sex chromosomes fail to separate. Okay. When sex chromosomes like XX or XY, generally they have to be separated. They have to be subjected to disjunction. Okay, before going into the gametes, they are to be separated. But if they fail to separate, they enter into the gametes collectively. Combinedly, they go into the gametes, and that will cause the production of sexual aneuploidy individuals like Klinefelter syndrome or Turner syndrome. Let us see here what happens. So mother has 44 autosomes XX. What are the gametes of mother? Let us see. So these two X chromosomes fail to separate, so that the two X chromosomes collectively go into one egg. So one egg here, instead of having only one X, two X chromosomes are there. And you see the other egg, there is no X chromosome. And you see the father sperms are normal, healthy sperms. Then you see the egg with two X chromosomes, if it is fertilized with the sperm with X chromosome, triplex females are born. Triplex females are, they look like females, and they are very tall in nature and with certain distinctive features. And you see, if egg without X chromosome is fertilized with sperm with X chromosome, then XO, Turner's syndrome, is born. And if the egg with two X chromosomes is fertilized with the sperm with Y chromosome, then XXY, the Klinefelter syndrome, is produced. So the production of triplex females, Turner's syndrome, Klinefelter syndrome, is because of only one reason, that is non-disjunction, which is nothing but failure of separation of the two X chromosomes. Okay. So what is Klinefelter syndrome? 44 autosomes plus XXY, total 44, 45, 46, 47 chromosomes. Okay. Here Turner syndrome, 44 plus only 1, that is 45. Only 45 chromosomes. Actually 2n number is 46. This is 2n minus 1. Here 2n plus 1. Here 2n plus 1. These are called sexual aneuploidies or sexual anomalies, we can say. These are the references. Okay. Thank you, friends. Thank you for watching this video. Hope you have understood the process of Correns theory of sex determination. Thank you.